Okay, we are live now and good to go.
Okay, I'm gonna let them in.
Well, hello everyone. Good morning, good evening, and good afternoon. Today we are going to have a webinar series. We'll wait a couple minutes for people to come in. We probably started 5 past the starting time. So just settle in and enjoy the webinar in a minute. So hello everyone, welcome to the AmeriFlux webinar series. Today we're going to have an early career scientist spotlight. We'll wait a couple minutes for people to come in, settle in. Okay, again, welcome everyone to the AmeriFlux webinar series. So today we are going to have an early career scientist spotlight, and we have a roster here on the screen and also show on the webcam here. And we just some ground rule about today's webinar. So we have 4 presenters today, and each one of them will have 10 minutes to give their presentation. And by the end of each presentation, we probably have a very short, brief Q&A. So just if anything is missing or not quite understand, and you can always put your question in the chat here. And if it's just a short question, and if you want, if we have time, and you can always welcome to raise your hand, and we can let you unmute yourself and to ask the question. Okay, and by the end, we probably have a 10 to 15 minutes for open questions. So there will be plenty of time for everyone to asking question and make a comment or any suggestion. Okay. So I'm going to introduce today's first speaker. Just a minute. So our first speaker is Bhaskar Mitra. So Bhaskar is from Northern Arizona University. So the floor is yours, Bhaskar.
Thanks, Jason. Can you hear me? Yeah.
Yes.
Okay. Hello, everyone. Thanks for attending my talk. At the very onset, I would like to first acknowledge my supervisors and colleagues and my different funding agencies. There are 3 key themes of this talk. One will be focusing on the variability of CO2 and CH4 fluxes. Then the second one, analyzing the controls of CH4 and CO2 using spectral analysis. And then third one will be showing initial results of gap-filling methane fluxes using spectral filtering and machine learning algorithms. Now, next slide. And First, let's introduce the site. This is located in the lower coastal plains of North Carolina. These sites are registered in the AmeriFlux database, and there are 4 sites: 2 post-harvest and a newly planted loblolly pine plantation, approximately 8 years of age; a matured rotation age loblolly plantation, approximately 30 years; and finally a natural undrained bottomland hardwood forest, which is approximately 100 years of age. And our research site is focused on understanding this coastal freshwater wetlands response to climate, land use, and sea level rise. Next slide. Next slide. Now, let's me focus on the first results. Let me focus you on the figures on the left. Panel A shows you the cumulative NEE budget for 11 years and with the uncertainty, and it compares those annual budget with results from similar coastal freshwater wetland sites across the world. Panel B shows the cumulative methane budget for 9 years, and it compares those results with the similar values from coastal freshwater wetland sites across the world. There are couple of take-home messages from these results. First, from the panel B, we can see that the CH for flux is of high magnitude and 5-fold interannual variability. These values are greater than 95% of the wetlands, including tropical wetlands in Indonesia and Amazon. And most importantly, the CH4 and CO2 fluxes do not have an inverse relationship, which begs the question why. And I'm saying disturbance. What I mean by disturbance is, if to think about disturbance, you have to think where the site is. The site is located in the Pamlico Albemarle Peninsula, which is projected to be completely inundated by the turn of the century. The site is in North Carolina, which has experienced some of the highest rates of sea level rise across the world, with the accelerating wave action and storm surge, with saltwater intrusion converting healthy vegetation to dead trees. So what we are, as popular known as ghost forests. So in short, our site is really spewing out, is really a carbon hotspot. And these results have been confirmed by a recent study by NASA, which we were co-authors also, that definitely this is a site with extremely high values of methane and carbon coming out from the site. So when I say disturbance, I mean disturbance from the perspective of climate change. Next, in the next slide, I will be focusing on the CH4 and soil respiration time series. Now, both of these greenhouse gases are basically multivariate, multidimensional. They're important metrics of ecosystem metabolism, and there are controls and mechanisms which drive these fluxes at different multiple scale. To name a few: microbial decomposition, substrate control, phenology, soil water table depth. A spectral analysis of this time series shows significant peaks at different resolution, highlighting the fact that which kinds of brings home the idea of challenges, highlights the challenge of disentangling the confounding effects of these various lagged effects. To understand these various controls, what I've done in the next slide, I have shown a spectral analysis of soil respiration. On the figure on your left shows how a traditional approach of using a conventional temperature response function does a very good job of a fit. You can see a good R-square value. But in the lower panel on the left-hand side, what I've done is a residual analysis, and instead of using a conventional lumped analysis, I've done a frequency domain and error analysis, clearly showing peaks at different resolution. To understand those error better, I've done a co-spectral analysis between soil respiration and temperature, between soil respiration and moisture, and soil respiration and PAR. PAR being used as a substitute for GPP, and we see significant peaks at different temporal resolution, suggesting again that there are the confounding effects of these various effects of various drivers driving methane soil respiration at different temporal resolution. This study has been highlighted recently in this JGR paper, as I've shown in the lower line. Now, in the next slide, what I've shown a similar analysis for methane fluxes, where I've shown co-spectral analysis between methane GPP, and the blue region is the statistically significant regions: methane and LE, methane and water table depth, methane and atmospheric pressure, and methane and soil temperature. And we see high significant peaks at the diurnal control between methane and GPP and methane and LE, and some weak controls at the synoptic scale and seasonal scale between atmospheric pressure and methane atmospheric pressure and water table depth. Again, this study has been highlighted in an ag forest meteorology paper. In the next slide, the take-home messages from those two earlier papers is, what does this different analysis mean? what we're suggesting here, what we are proposing here is a spectrally truthful model. What I would means a model that would consider the dynamics of appropriate scale. And what we're suggesting here, proposing an idea of temporal scale dependence of different driving variables to simulate the ecosystem processes, which currently is not done. For CH4 and for RSS, we see GPP and LE is driving the fluxes at daily scale, temperature at daily and monthly, and pressure water table at synoptic and monthly. Similar thing we see for soil respiration. So this is a theoretical model which we are proposing, and using this model as an algorithm, we are trying to gap fill the methane fluxes as a case study we've done for soil respiration, but I'm just showing you for methane fluxes. In the next slide, I want to highlight first the panel on your right, which shows the methane time series, which shows the power, which shows the soil temperature, and which shows the water table depth time series, basically. And you can see the methane time series has gaps lengths of different, the gaps have different lengths, short gaps, small gaps. But in the lower three panels, I'm showing you two time series. One is the absolute time series, which is in red, and then is the spectrally filtered time series. Spectrally filtered in the sense that what I've done is a wavelet multi-resolution analysis, and I've kind of extracted for power the time series at the dial scale, for soil temperature the time series at synoptic scale, and for water table the time series at the seasonal scale. And I've also added the wavelet smooth vector. So instead of using the red, I'm using the blue time series, and I'm gap filling this using this 3 input data. I'm gap filling methane using a machine learning algorithms, which is a spectral gap filling approach. And I'm comparing them with the conventional nonlinear regression approach, where the input data is soil temperature. If you can see on the panel on your right, on the top right, left-hand corner. Now, for the nonlinear regression approach, I'm using an OLS algorithm, which is ordinary least squares, which assumes the data has to be normally distributed. But in a machine, and for machine learning algorithms, which does a better job in capturing nonlinearities and doesn't make any underlying assumption about whether data is normally distributed or not, we are testing different algorithms. These different algorithms, again, I'm not going into details, but they are different. They test this, but I'm trying this different algorithm using different, and going by using gap filling the methane fluxes using these 3 spectrally filtered time series. Now, to do an uncertainty analysis, I've introduced artificial gaps in the methane time series, and I've done this analysis 20 times, and I'm summarizing the result using the summary of the 20 results, and I'm highlighting them in the next slide. In the lower top left-hand corner panel, so what I've done is I've partitioned the methane time series into training set and testing set. 70% of the data was used to train, 30% of the data was used to test. Now, I've done this 20 times, so you can see the summary of those 20 results using box plots. And you can see the red is the testing. Testing is the known data, and the importance is the training, how the model behaves on the unknown data, the trained data. For the different machine learning algorithms, one is elastic net, one is the radial kernel support vector machine, one is random forest, one is the XGBoost, one is the artificial neural network. For random forest, we see the lowest RMSE and that kind of gives you, and lower the RMSE, better the model performance, because that's the metric I'm using. So the panel on the left-hand side summarizes that result. But the big question is, since I'm proposing a new gap filling approach, how does this behave with the conventional nonlinear regression approach? To do that, I'm using a metric called an F square, and you can see the formula. And the B on the denominator of RMSE is the base model. And F square, higher the F square, better the model performance. And we can see the best model performance is for random forest. Now, since I've identified the best random forest per algorithm doing the best performance, I've kind of done a one-to-one fit on the lower left-hand corner panel figure. And you can see the random forest algorithm does a better job than a very conventional temperature response, nonlinear temperature response function. Finally, in summarizing my results in the next slide, the big picture here, year-to-year variability of methane is not inversely related to NEE. We are suggesting increasing hydroperiod and tree mortality are expecting to lower the GPP and having a lower priming effect on methane. Spectral footprints of methane and soil respiration driven by power spectra of superimposed environmental and biological forcing. Methane and CH4 varies diurnally driven by substrate availability. And our initial spectral gap filling results highlight improvement over conventional temperature response function. Finally, I've highlighted a couple of other papers where our carbon dynamics story has been highlighted. And if you have any questions about the site, please feel free to ask Dr. Nurmitz or Dr. King. If you have any questions about some of the methodologies I've adopted, please feel free to email me. Thanks.
Well, thank you, Bhaskar. I think we have time for one short question, just very brief one. Am I going to bring up the presentation for the next presenter too? If no, I have a short question for you, Bhaskar.
Yeah.
So have you compared, you're using the wavelet decompose time series to do the modeling. Have you compared, if you're using the original time series to do the modeling, how much you actually improve that?
Yes, we have done that. So you're saying instead of using the original time series, if comparing the original time series and spectrally filtered, whether there's an improvement or not?
Yeah, just compare just how much it gets.
Some of the algorithms didn't do much improvement, but the random forest did. So not all the algorithms. So like the elastic net regression approach, which takes into consider multiclonality, didn't improve much, but the random forest wasn't improved compared to the original time series.
Okay, thank you. I think people can maybe come back, have a question in the later discussion. So our next speaker will be Lisa Haber. So Lisa is a PhD student from the Virginia Commonwealth University. So the floor is yours, Lisa.
Thank you. I just saw a little notification that my internet is unstable, so hopefully there won't be any issues here, but I apologize in advance if there are. Yeah, so I'm really glad to be here today, and I'll be telling you about my dissertation research. There's sort of two arms of my work in graduate school, and each of them is a set of studies located at an AmeriFlux tower site. So I thought, even though this is a very descriptive and introductory presentation as opposed to very data-driven, that it might be of interest to you all, and I hope so. Next slide, please. So just briefly, a little background on me. I am a student of Chris Goffs at Virginia Commonwealth University. Our group is interested in both above and below ground carbon cycling response to disturbance in different landscapes, primarily in northern temperate forests. I have really enjoyed becoming more connected with the AmeriFlux network over the last few months, attended the fall meeting last fall, and am now on the DEI committee along with André and Christine and others. In a former life, I was a high school teacher, so I'm very interested in science education and communication in addition to research. And I also have an almost 2-year-old. So that's a little about me. Next slide, please. So as I mentioned, 2 arms of my dissertation are taking place at AmeriFlux sites. Only one is really going to use flux data, but they're both at network sites. So hopefully these will be of interest to you. One is up here in northern Michigan, where I am at the moment at the University of Michigan Biological Station. This is an AmeriFlux core site, and there's 2 towers up here. And then the second is a recent addition to the AmeriFlux network, a tower that was built in 2015 in the coastal plain of Virginia at the VCU Rice Rivers Center. But across both sites and studies, I have an interest in ecosystem carbon cycling in response to disturbance, and really interested in leaf functional response and how that scales up to ecosystem functions such as production. Interested in leaf functional traits, both as a predictor of ecosystem response and also a response to disturbance, depending on which sort of lens you take. Next slide, please. So where I am at the moment, up here in northern Michigan, there are 2 AmeriFlux towers. They've been running for a long time, collecting data here. So this is in the Great Lakes ecoregion. The USUMD tower is located at an earlier disturbance experiment at this site called FACET. That's the Forest Accelerated Succession Experiment, started in 2008. And the current disturbance experiment that's running, that my dissertation research is coming from, I'll describe in just a moment, but it's really sort of an outgrowth of this earlier experiment where early successional trees were targeted for experimental mortality, and carbon cycling was tracked following that. Next slide, please. So the Forest Resilience Threshold Experiment, or FORTE, was initiated in 2019. This involved stem girdling, which is cutting a ring of bark and phloem tissue off of the sort of base of the tree and killing it. That took place over 8 hectares across the landscape up here, killed about 3,800 trees. Some are still in the process of dying. So this year, we anticipate sort of being peak disturbance when the trees that are still hanging on finally succumb and senesce this year. And so the idea here is to track carbon cycling response to disturbance across this replicated gradient of severity from 0 to 85% gross defoliation of the plot. So just to orient you on this map, the 2 little tower illustrations here, the one on the left in green is USUMB. The one on the right is USUMD, or the Facet site, and that's the earlier experimental disturbance that I mentioned. So FORTE is really nested within the footprint of USUMB, takes place, as I said, over this replicated disturbance severity gradient. The idea there is one of the major findings in the earlier experiment in Facet had been that up until about 65% LAI loss, the forest really didn't see an ecosystem productivity decline. So there was sort of sustained production following disturbance up until that threshold of leaf area loss. So one of the ideas in FORTE is to really get at the mechanistic underpinnings of stability in this ecosystem. All of the data that we've been collecting and a whole suite of structural metrics, forest production, we have leaf level physiology and soil respiration, all kinds of things available through the R package FORTE data. Next slide, please. So my little chunk of this is looking at, as I mentioned, I'm interested in leaf function and leaf physiology. So tracking physiological, morphological, and biochemical responses at the leaf level following disturbance, and trying to figure out where the stability declines across the severity gradient. So this flow chart on the bottom kind of illustrates my thought process here. So disturbance is going to shake up and change ecosystem structure, allow for resource availability to surviving plants. That should precipitate some functional response at the leaf level that maybe is scalable to an ecosystem metric like NPP. And I'm getting at this with several different instruments. And also on the left, you see a cherry picker there. I've been taking leaf samples in both the canopy and subcanopy strata. So I'm interested in how those different strata of the forest may be responding differently. And in the subcanopy, really interested in how those released trees are able to take up and use newly available resources. Next slide, please. Okay, so I mentioned this is very light on data. This is a very preliminary couple of figures here, but just showing where I'm going to be looking and working this fall. So on the left-hand side here is a set of box plots showing community-weighted leaf photosynthetic rate across subcanopy plants that I've been sampling for the last 3 years, and showing how the community-weighted mean leaf photosynthesis is changing over time and potentially in response to increased disturbance severity. So in black, we have our control community-weighted mean across the landscape, and then green, blue, and orange are the 3 different disturbance severities. So there's clearly large interannual variability in leaf photosynthetic rate, which is not surprising. Wetter, cooler years, or years with greater soil moisture will have higher leaf photosynthetic rate. But we are potentially seeing a response to disturbance severity that I'll be getting at with some much more rigorous analysis this fall. On the right-hand side is the ecosystem production metric that I'm interested in for this particular study, looking at the subcanopy. So I've got wood net primary production. This is just 2020 values, but it sort of illustrates here how we're seeing maybe a compensatory effect in the subcanopy as canopy trees are dying due to the girdling. The subcanopy may be sort of picking up the slack and sustaining production, at least up to a point. Next slide, please. Okay, so wanted to round out here. That was very like mile wide, inch deep introduction to some things going on in Northern Michigan, but this may actually be of greater interest to the Flux community since these will be newly available Flux data coming from this site in the coastal plain of Virginia. This is USRRC for the Rice Rivers Center at BCU. And this is a really, I think, a unique site in terms of the different descriptors of it in the AmeriFlux network. This is both a restored wetland and a tidal freshwater system. So if there are multiple of those, there are not many. But this is a 70-acre wetland site in Virginia that has tidal communication with the lower James River. So it is freshwater, but tidally influenced. So that's kind of interesting. We have carbon dioxide and methane sensors located out there. Due to a lack of sustained funding and graduate student allocation of time, we have not had a continuous data stream available from this site yet, but we are anticipating that that will be corrected beginning this fall. And in addition to those Flux data, we expect to be able to collect continuous water table height, soil data, the typical meteorological and PAR data, also dissolved nitrate, salinity, conductivity, canopy height, and greenness. So my interest here is to just characterize the principal biophysical drivers of carbon cycling in this system, since it is a relatively understudied type of wetland. But there are clearly going to be a number of data types that may be of interest to different members of the Flux community. Lots of questions that could be asked here over time. Okay, I think I'm right at 10 minutes, so I'll go ahead and end there. Final slide just asks you for questions, and thank you.
Well, thank you, Lisa. I think we probably need to move forward to the next one. But people can come back and maybe have a question later during the Q&A, this open discussion. Just give me a minute. Okay, our next speaker is André Santos. So André is from Federal University of Panama, Brazil. So André, you're on.
Hello, my name is André Santos. I am an early PhD environment engineer from the graduate program Environment Engineering at the Federal University of Paraná in Brazil. And also now I am a member of the FluxNet Early Career Network organizing the next slide, please. And can you go to the next, please? So as our background, and in my graduate studies, I worked in the environmental monitoring and modeling studies laboratory, and specifically in two research projects. The first one is the GS Verde, that is to develop methods for measuring greenhouse gases fluxes, and a case study for the Green River Reservoir. And the second research project that is specifically linked to my dissertation is a project for development and application of numerical methods for the analysis of environmental problems. Next slide, please. So in my master's, I used a IVAC model from our laboratory, a model that was used to study the influence of the Itaipu Lake on the regional climate. And the model is based on the mathematical model, but the model was totally coded in Fortran here in our laboratory. So firstly, I used this model that estimates only water vapor fluxes. And I implemented a model for estimating also the CO2 flux. And then I changed this original stomatoconductance model for a more physiological one. And I calibrated the two models concerning sensible and latent heat flux in a soybean crop in Brazil from our laboratory. And in this field campaign, we don't have CO2 flux measurements. but the two models have similar performances for estimating the sensible heat and latent heat, however, different responses for CO2 flux. So we went to do another comparison in a soybean crop in Argentina that had CO2 flux, where we found that the SWAT model to the physiological model had better performance, but we found a poor representation of the nocturnal flux. And next slide, please. So in my doctorate, I tried to study the dynamics of respiration processes. And next slide, please.
Andre, are you able to
Yes.
get a little closer to your microphone?
Okay, sorry. It's good now.
It's better.
So, first I tried to study the leaf respiration. So in Campo de Mayo, Argentina, we did a short-term simulation campaign where the leaf area index, the fraction of vegetation covering the soil and the plant height were constant, were calibrated in the model. And we used soil respiration, the original soil ecosystem respiration of Calp. So in the graph above is the leaf respiration, and the negative flux is the flux of CO2 from the plant to atmosphere, and the positive from the atmosphere to the plant. So on the two graphics below is showing the total CO2 flux, where on the left is the total CO2 flux, on the right is the zoom on the night periods. And we tested a case without leaf respiration and another four respiration formulations. And we tried a correlation with local data where we didn't obtain success. Next slide, please. And here we found that the SWAT model with the better result for the total CO2 flux had the worst result for the nighttime period. But we diagnosed that all the respiration formulations have influence of the temperature. We found a better prediction of total CO2 flux than dark flux, and we suppose that respiration is well understood and more difficult to predict than photosynthesis. Most of the respiration calculations, it's a primary order decay function from temperature. And we also found some limitations of the eddy coverance technique in low turbulent activity for comparison and calibration. So at this point, we decided to partioning the CO2 flux into more parcels. And next slide, please. So here in Mercedes, in a city of Mercedes in Argentina, another field campaign, we have the 3 short-term simulation campaigns, one on the plant growth. This is all for soybean. One short period for growth, for full development of the plant, and another for senescence. And so we tested the three formulations for leaf respiration of the active biomass that are in blue. In yellow are 2 structural respiration, and in red is a total ecosystem respiration formulation. And we tested the combinations of this. For this, we implemented a plant growth modeling. So the plant do photosynthesis and absorb CO2, growth biomass, and he allocate the active biomass for structural biomass compartment above the soil and below the soil, and then we calculate the respiration of the structural biomass. And here we the leaf area index, the VAG, and the plant height was varying during the plantation, during the crop, from remote sensing data. Next slide, please. So here we have the leaf respiration. On the left charts are the growth period, and the right charts are the senescence period. And they look similar, but there are different combinations of leaf respiration with structural respiration and with soil respiration. And as we can see here, we have some respiration. Some leaf respiration models have different compartments. The purple line shows that the leaf respiration smaller on the senescence than on the growth period that we should expect. But the blue model have a different response. It calculates higher leaf respiration on senescence than on the growth period. Next slide, please. And here are the soil respiration combinations that we tested, and I highlight with yellow because are different scales. So as the four charts below, as expected, the structural respirations are higher in the senescence than in the growth period. But in the total ecosystem origin of the ISBA model, the total ecosystem respiration are smaller on the senescence than on the growth periods. And next slide, please. And here is the total CO2 flux, where we changing the respiration formulations. We can see some differences on the nighttime period, but at daytime they are not influenced too much on the growth period, but on the senescence period, also the respiration formulations influence on the daytime CO2 flux. And next slide, please. So here I cannot show all the results from soil temperature, humidity, and biomass growth, but what we found is that leaf respiration interacts with vegetation growth and with soil moisture, and as the soil respiration, the structural respiration does not. This was expected, and the colloid's leaf respiration accounts for more than only the active biomass respiration. And two of the leaf respiration are sensitive to stomato from mesophyll conductance for water, and another the colloid one is sensitive to air temperature. So after rains period, different formulations has different responses. Our soil one, the original account for a global ecosystem respiration, and the formulation the 2 and 3 soil respirations account for plant structure, but we're still missing some heterotrophic respiration. Next slide, please. And so, and as this, we had some problems with this data set from Mercedes. We could only extract it at 3 periods of data set with quality to, so we decided to go to AmeriFlux. This is what, when I know I met AmeriFlux, and we find this data set on Nebraska with a long term from 2002 to 2000. It's a long-term campaign with soybean and maize, and from here we have they have also biological auxiliary data sets with leaf area index and plant height measured at the field. So we decided to also implement a dynamization of the model. So here we did empirically estimate correlations between leaf area index and biomass, and plant height and biomass. So now in this version of the model, the model start evolving interactively. So we don't need any, in this version, we are not using remote sensing data for input. The leaf area index and the plant height evolve as the plant absorbs CO2. And next slide, please.
André, you want to wrap up quickly? Okay.
And so this is the from this Nebraska data set. On the first chart above is the point are the biomass, active biomass. The green line is the active biomass calculated by the model. The blue line is the total biomass, and the yellow one is the structural biomass. In the middle chart is the leaf area index, where the points are always the data and the lines are the model. Here we have some discrepancies, but the model was calibrated with, on the previous stage with remote sensing data. And here in Nebraska, we are using field measure data. So we have some discrepancies. We need to maybe calibrate it again. But for the plant height, the results were very promising. So from the next step for the article of my doctoral thesis, I want to test more heterotrophic respiration to complete the CO2 balance of the nighttime period. I am coding an automatic calibration because I did all the calibrations manually, and I want to take this bias out of the calibration, and to do the journal submission. And I'm looking for a postdoc position. And next slide. It's just I'm just showing other projects that I work with other colleagues here. Dynamic is a software that we developed to study gravity currents, and the size sensor is a project that we developed low cost sensors. And here on the right, we are testing for turbidity. And last slide. Thank you for the attention.
Well, thank you, Andre. I think we need to move on to another speaker, but we'll come back if people have any question for your slide. So our last speaker will be, just give me a second. So our last speaker is Stephen Kannenberg from University of Utah. Steve, the floor is yours.
Great, thanks. Well, yeah, thanks to all of you for being here, and for to AmeriFlux for putting this on, and all the other speakers today who had some really interesting talks. So yeah, I'm Steve Kannenberg. I'm a postdoc at the University of Utah, and a lot of what my research is centered around is understanding what happens to forests during drought stress, and a particular focus of mine is what happens following drought. So are trees able to recover from drought, or are they put on this multi-year trajectory towards their inevitable death? And today I'm going to talk a little bit and kind of tell you a story about the latter, which is when trees die, and how we've been merging eddy covariance with some physiological and modeling techniques to try and understand a recent drought-induced die-off event in southern Utah, which you can see just a snapshot of here in the background of this title slide. But this really isn't your run-of-the-mill mortality event. It's actually quite puzzling from a scientific perspective. So let me explain why really briefly here. You can go to the next slide, please. Cool, yeah. So this is a picture of southern Utah where I do a lot of my research, and it's very apparent from this picture that this region and a lot of the American Southwest in general is extremely dry. You can hit the animation thing there. And unfortunately, this region is also getting much drier. There was a recent study using tree ring chronologies, or reconstructions of soil moisture using tree ring chronologies, that found that in the southwestern region, as like kind of in the aggregate, we've been in this 20-year now mega drought is the term they gave it. And it's been the second driest period in the last 1,200 years. Next slide, please. So this ongoing aridification, this mega drought, whatever you want to call it, has some pretty drastic consequences for the dominant ecosystem type in the region, which are pinyon juniper woodlands. It's this incredibly widespread ecosystem type consisting of pinyon pine trees and juniper trees that spans a lot of the American Southwest, as you can see in that graph there. Next, please. So sure enough, we're kind of at the start of this mega drought period. We had a couple of really bad droughts come through and kill off a ton of pinyon pines, which is what you can see in this picture here. Almost all of those dead trees that you see are pinyon pines. And these drought-induced tree mortality events of 20 years ago have really spurred on a ton of research as to the ecology and the physiology of both pinyon and juniper species. And to make a really long and really rich literature on this topic kind of short here, it makes a lot of sense why pinyon pine dies following drought, because for a number of reasons, it's much more susceptible to drought than these co-occurring juniper species. Next slide, please. Great. So now comes 2018, which is actually when I moved to Utah and started my postdoc. We kind of got put into this extremely severe drought. And in some regards, it was probably more severe of a drought period than those droughts we saw around 20 years ago. You can see here during the WADA year of 2018, we had lower annual precipitation and a higher mean vapor pressure deficit than any of those individual years that came in or any of those individual years from 20 years ago that ended up killing off a ton of trees. Next, please. But what's puzzling about this one is this time it's the juniper species that are dying, and most opinions are actually pretty okay. I thought this picture is a really good diagram of just, you know, how puzzling this is from a scientific perspective and just how odd this event is. Junipers are some of the most drought tolerant trees, or at least the ones in the southwestern United States that we can measure. They're incredibly resistant to embolism, so they can tolerate extremely negative pressures in their xylem. There are really no insects or diseases that are known to sweep through and be able to kill these things outright. These things are hundreds of years old, over 1,000 years old in many cases, and they've done just fine until recently. And they've died really, really quickly too. This picture was taken, and all the ones in this talk were taken in the fall of 2018 when the drought was really just starting to let up a little bit. And you know, they've done some experiments reducing water to, you know, these ecosystems, some like rain out shelter type of things. And in those experiments, it's really taken multiple years for these trees to start losing some of their foliage. And we saw these just within a couple of months. Next slide. So, you know, we were really kind of saddened by this mortality event as we were observing it happen, but also a little bit excited from a scientific perspective because it is such an odd event. We really wanted to figure out what exactly is killing these things, because the speed and severity of this is probably unprecedented in the region, at least in recent history. And finally, we wanted to, you know, answer the crucial question here: is this just a one-off random tree mortality event, or is it kind of an indicator of a pretty grim future for this incredibly widespread, important pinyon juniper woodland ecosystem? Next slide. Cool. So we're tackling this from a number of different perspectives, but I wanted to highlight what would probably be most interesting to the AmeriFlux community, which, if you'll hit the slide, put too many animations in this one, is our eddy covariance tower that we installed kind of in the heart of this dieback region. Actually, this is a rapid response system that's on loan from the AmeriFlux Management Project, and it's really given us a fantastic opportunity to pair ecophysiological measurements and some plant hydraulics modeling with these really important eddy covariance data. Next slide, please. So the goal, what we're trying to do here by pairing the flux data with all of these physiological measurements, is to simulate past, present, and future health of these woodland ecosystems. And this is really going to let us test some key hypotheses regarding why these trees are dying. One of the hypotheses that we have so far, that actually so far, even without the eddy covariance stuff, has a good amount of evidence behind it, is that we've seen huge reductions in winter precipitation and deep water stores, which are very important for these juniper trees, and we can test the consequences of that across the whole landscape with this model as well. Using this physiological eddy covariance modeling trifecta, we can also ask some really neat questions about, you know, what's the degree of plasticity or adjustment of the physiology and allometry of these trees that will allow them to survive as this region inevitably only gets warmer and drier. So can they just shed some of their needles and be fine and survive? And of course here, you know, the broadest, I think most important reason for this research is just to understand, is more dieback going to occur in this region? Next slide, please. So we might be a little bit closer to answering this question than we initially thought when we started this research in 2019, because this is the current, or just a couple weeks old, US drought monitor for the western half of the United States. And you can see that that a ton of the western United States, especially the southwestern region, is in really, really bad drought right now. And just for comparison, if you'll advance the slide, this was what it looked like in August of 2018. So we saw a little bit of juniper dieback right in that Four Corners region pocket of D4 drought, but now that's across pretty much the entire range of pinyon juniper woodlands in the country. So you can go to the next slide. So the answer to this question here, is more dieback going to occur, is likely yes. And sure enough, we've received a bunch of reports of a lot of juniper trees dying, a ton of them in northern Arizona, all across all across Utah, a little bit in Nevada, and some in Southwest Colorado as well. So it seems like what we saw following this 2018 drought was kind of just the tip of the iceberg for tree mortality in this ecosystem. Next slide, please. So luckily, because we had that drought in 2018 and we set up all of this research infrastructure, including the tower here, we're really well positioned to monitor this drought as it develops, partially because we have this eddy covariance tower. It has a rain gauge. You can measure all the fluxes of carbon and water in and out of the ecosystem. And we've got this depth profile of soil moisture probes down to bedrock. So we can really characterize the severity of this drought and its impact on carbon and water fluxes. But if you advance the slide, please. We also, just a couple months ago, installed 14 instruments called stem psychrometers on these juniper trees in the tower footprint. And these instruments allow us to semi-continuously monitor the hydraulic health of these juniper trees. They monitor water potential semi-continuously. And this is going to allow us to track water as it moves through the ecosystem from precipitation to percolation through the soil profile, through these trees and out to the atmosphere in terms of ET. And it's going to be a really cool way to monitor how this ongoing drought is impacting juniper health. And once again, something that's kind of sad but interesting from a scientific perspective is even these trees that we instrumented with these sensors, they're starting to die back a little bit just in the last month here. So we might get a really neat tree-centric and ecosystem-centric perspective on what happens during one of these dieback events. So you can go to the last slide, please. So just to conclude here, this normally extremely drought-tolerant juniper species has kind of been pushed to a threshold due both to the presence of this 20-year-long mega drought period and these more severe acute droughts that have happened recently, and the future of this ecosystem across the entire United States Southwest is really uncertain. I think the final message here is just that pairing eddy covariance with physiology and modeling techniques, at least for us, has been a really interesting and really crucial step towards understanding the mechanisms behind this dieback and how this landscape might change in the future. So that's it, and I'm happy to take any questions.
Well, thank you, Steve. Any quick question for Steve before we move on to the open floor?
I have one. It's kind of general. Steve, are there any more drought-resistant species than juniper in this system or in the United States more broadly?
Not really in this system. It's not all that diverse in terms of tree species of an ecosystem. In the United States, maybe it depends on how you really define it, but juniper has some of the most resistant to xylem embolism around, and it generally has extremely deep roots. So that's kind of why it's regarded as incredibly drought tolerant species. There might be one out there that in some regards is more drought tolerant, but if it's not the most drought tolerant, it's one of them.
So that's really, really alarming.
Yeah.
Okay, thank you again. I'm going to stop sharing my screen. So I think we have a couple minutes, 6-7 minutes to open the floor. So if anyone have a question for any of the speaker today, now is the time to bring up. And
I have one. If this is one of the last things that Steve was mentioning. It just got me curious about that instrument that you have attached to the stem. Can you tell us a little bit more about that?
Yeah. So as I mentioned, they're called stem psychrometers, and what they do is you kind of expose a little piece of the xylem of the tree to this It's just a pair of thermocouples in kind of a sealed chamber, and it uses a little cooling pulse to kind of, it equilibrates with xylem water, use a little cooling pulse just to kind of back out the water potential of the air that's equilibrated with the water potential in the xylem. And it's a really cool tool for the approach that we're using here. It's the instruments are a little bit nitpicky. They require a lot of cleaning and calibration, and so I think they haven't been as used as much in the past as for kind of ecology, forest ecology techniques as you might want. But we've got 14 of them, and hopefully they stay running for the entire summer.
Sounds cool. Thank you.
So Dennis, do you have a question for the speaker?
Yeah, good morning. Great job, you all. I always like listening to everybody. I have a philosophical question maybe for the group of you all as you start comparing treatments and have paired flux towers or multiple flux towers. There's communities out there that like to criticize us about this issue of pseudo-replication. I'm just kind of curious if you guys have run across that, if you have any defenses to this other community, if you thought about it. Just kind of an open-ended question, but I think one we always should think about. I have my own biases, obviously, but.
That's a tough question. I don't know, Lisa, want to say something about that?
Sure. Yeah. So I mentioned here, this is not so much paired flux towers as just pseudo replication being sort of something you can't escape in ecosystem studies, right? So here in Forte, we have these 4 different replicates. We're replicating the disturbance treatment, but there are known differences before disturbance across this very small area, right? So microclimatic and soil variation is just inescapable. And it does matter. We're seeing we're seeing differences, yeah, both in terms of pre-disturbance variables and response. So I don't know. I guess I would respond that this is not a lab bench. It's the real world, and I don't think. I mean, there is value in comparison. It's absolutely necessary, but it's not possible to control for all of it. So I guess that's
a good answer. I think we just all should be prepared to think about it. The 2 things I guess I come back with is one is obviously we always want to think about flux footprints and see what we see, but I always like to think that our eddy fluxes are actually measuring a population, whereas this multiple sampling or subsampling that population. So I think that works to our advantage. So something to think about as we deal with these issues. So.
Steve, you want to say something about that? I think you're facing a similar situation.
Yeah, sorry, I was typing a response to John here. There. So the issue is just, I missed part of the question. I'm really sorry. I was trying to respond to John's question.
Oh, I'm sorry. No, we were asking about issues of pseudo-replication. Sometimes different communities criticize us that we only have 2 flux towers, but I guess the context is we're often measuring the whole population as opposed to subsampling the population as you would with smaller, older techniques, essentially.
Yeah, okay. That's, I mean, my take on it is I think similar to what Lisa said there is like, this is the real world. How we're doing this research is kind of shaped by the tools we have. And I think that, in a lot of cases, even if we could set up a perfect like experimental grid of flux towers, I think due to how the technique works, we wouldn't be able to have as clean of data as we might want in that regard. And so I think, partially getting around that, it really seems like having these networks of where you have really long running flux towers in similar ecosystem types that, you're not exactly replicating the same exact question, the same context within the same ecosystem, but when you start approaching similar questions from different angles using slightly different data, slightly different ecosystems, that type of thing, then I think that's one really interesting way that having this kind of networked approach for eddy covariance towers can help us maybe get past that issue.
This is the power of Bhaskar's expertise on time series too, is you can start teasing apart multivariate controls. And so I think this whole team today is a nice theme on how to deal with all these issues that are really interesting. That's where the science is going forward. Thanks.
Anyone have a question? I think we have a minute or two. Okay.
Steve, I have a quick question.
All right, go ahead.
How at what interval are you measuring those this water potential, like every hourly or?
It's half hourly, so it'll be directly paired with how we're processing the flux data, which is really neat.
That's cool. And they're okay.
Okay, I think we need to wrap up here, but people feel free to stay if you want to talk more about some of the very exciting idea about new research. So let's get a big round of applause to all our speaker, and thanks to presenting today. And most of them have to juggling with field work and family and some other issue at the same time, but it's good to see everyone here.
He said, thanks for navigating my talk. What to say?
It's not too difficult. Yes. And just a quick announcement. I think we are going to have another, the last young early career speaker coming up in a few weeks. This one will be a Spanish language webinar. So stay tuned. We'll send out an announcement soon. So thanks everyone to join today. I'll see you sometime soon.